Okay, this is part 2 in our 4-video series of predicting genes in a genomic DNA sequence. In this video, we will be learning about how to identify the true translational start of a protein coding open reading frame. Open reading frames that code for proteins have features that allow a ribosome to initiate translation at the start codon. Most protein coding genes have an ATG start codon, and this is because it most efficiently complexes with F-methionine tRNA that is the initiator tRNA for translation. The ribosome then reads in 3 base pair intervals, allowing new aminoacyl tRNAs to enter the ribosome and extend the peptide chain. Ultimately, protein synthesis will stop when the ribosome reaches the stop codon and dissembles, releasing the newly synthesized protein. Open reading frames can have any of 3 stop codons, TAG, TGA, and TAA. But bacteria and bacteriophage will on occasion use the start codon GTG, and even less frequently, the start codon TTG. These each, although less optimally, interact with F-met tRNA to initiate translation. If there are so many start codons and open reading frames within a single mRNA sequence, how does the ribosome know where to bind and initiate translation at the correct start codon? For example, in this gene model, there are 2 in-frame starts. What dictates where the ribosome will assemble and begin to translate? The 30S ribosome contains 16S ribosomal RNA that is complementary in sequence to the Schindel-Garneau sequence that is located just upstream of the start codon. The Schindel-Garneau sequence, therefore, recruits the ribosome to the correct start codon to initiate translation. Once the F-met tRNA and the 50S ribosomal subunit joins, the correct AUG start is aligned to the P cavity of the ribosome where the transpeptidation reactions occur. In E. coli, the Schindel-Garneau sequence is a purine-rich sequence, AGG, located 4 to 9 base pairs upstream of a gene's start codon, and is important for recruiting the ribosome to the gene start codon for translation initiation. In mycobacteria, the Schindel-Garneau sequence is also a purine-rich sequence that is positioned further away from the start, around 6 to 12 nucleotides upstream of the start codon, and may form 5 to 8 base pairs with the 3' end of the 16S ribosomal RNA, UCUUU, CCUCC. Often, 3 nucleotides in the Schindel-Garneau sequences, such as AGG, are enough to initiate translation. The sequence of the Schindel-Garneau sequence and its location relative to the start codon are important predictors for translation initiation efficiency. A start associated with a strong Schindel-Garneau sequences is likely to be translated more efficiently and is a good indicator of the correct translational start. What does a strong Schindel-Garneau sequence look like? A strong Schindel-Garneau sequence is close to the canonical mycobacterial sequence of AG, AAA, GG, AGG, and will anneal more efficiently to the complementary anti-Schindel-Garneau sequence within the 16S ribosomal RNA. The location of the Schindel-Garneau sequence relative to the start codon also contributes to the efficiency of ribosome binding. Within the ribosome, the anti-Scheindelgarno sequence of the 16S ribosomal RNA is a fixed distance from the P site. If the Scheindelgarno sequence is to efficiently anneal to the 16S ribosomal RNA and position the start codon within the P site of the 30S ribosome, the distance between the Scheindelgarno sequence and the start codon must be 6 to 12 base pairs in order to align with the positions of the 16S and the P site. We can use computer programs to measure how strong a Scheindelgarno sequence is by determining, first, how close the sequence is to the Scheindelgarno consensus sequence, and second, its distance from the start codon. The closer a sequence matches the model of the Scheindelgarno sequence, the better the score, with the highest scores, or the least negative scores, being the best scores. A strong Scheindelgarno sequence is a good indicator that we have identified a likely gene and also a likely start for that gene. But what if a putative gene does not have a strong Scheindelgarno sequence? Does that mean it's not a gene? It could mean that it is not a gene, but it also could mean that it is a gene that has either a weak Scheindelgarno sequence or a gene that lacks a Scheindelgarno sequence altogether. Genes that are expressed highly typically have strong Scheindelgarno sequences that allow more efficient translation initiation. But some genes are expressed at lower levels and are likely to have weaker Scheindelgarno sequences that allow less efficient translation initiation. And some genes lack a Scheindelgarno sequence altogether. At least half of mycobacterial transcripts are either leaderless or have a 5' untranslated region that lack a Scheindelgarno sequence. Translation initiation of these transcripts requires ribosomes with alternative compositions than the canonical 70S ribosome. In other words, translation initiation occurs by alternative mechanisms. The strength of the Scheindelgarno sequence is also impacted by operon structure. Genes located at the start of a transcript, such as single genes or genes positioned at the beginning of operons, are likely to have strong Scheindelgarno sequences. Most genes in a phage genome are expressed from polycistrons or operons. These are groups of protein-coding genes that are expressed on a single mRNA from a single promoter. Sometimes there are very small gaps between genes in an operon, and there may be a Scheindelgarno sequence in these intergenic regions that allow translation to reinitiate in between open reading frames. But some open reading frames have weak Scheindelgarno sequences or lack them altogether if they are positioned with 1 or 4 base pair overlaps within the upstream open reading frame. Note that the 4 base pair overlap includes the stop codon of the blue upstream gene, UGA, which would cause a 70S ribosome to release the newly synthesized protein from open reading frame 1. If the ribosome shifts back a single nucleotide, it is now positioned to read the start codon of the orange open reading frame, AUG. The protein for open reading frame 2 is synthesized by the same ribosome without needing to disassemble and reassemble to initiate translation at this start site.